Let's take a look at each of the filter. Remember, by clicking on the icon at the top right of any filter, one can view or edit the setting for that particular filter. Let's now take a closer look at the genetic analysis filter. This filter allows you to identify variants that are consistent with a particular inheritance model, or that are present in certain percentage of case or control in a study. It is used to filter for specific genotype, differentiate one set of sample from another, for example, disease versus normal, affected versus unaffected, responder versus non-responder, or can be used to filter or test mode of inheritance. Whether control sample are available or not, this filter is the main filter for filtering on genotype and inheritance model. To help you getting started, there are several presets that allow you to configure the genetic analysis filter quickly for some of the most common analysis purposes. Depending on what selection you made when creating your analysis, a particular preset and default setting will be applied. This preset is somewhat of a best guess, and it is strongly recommended that you consider these settings carefully to be sure they match your particular study, assumption, and model. By selecting Set, the filter automatically adjusts itself to the preset selected. When two samples are uploaded into variant analysis with the identical subject identifier, the application will pair those samples. This is most common for tumor, normal, for identifying somatic variant on a subject-by-subject basis. The next option allows you to restrict variant based on the inheritance pattern. This option allows you to filter based on the pedigree file for a relationship between parents and children with Mendelian inheritance consistency across all parent-child genotype. Transmitted is a relaxed filtering of variant which implement minimal inheritance filtering and allow you for some sequencing and variant calling error. De novo specifically filter for variants that are inconsistent with parent-child Mendelian inheritance. Finally, Mendelian filter for variants that obey Mendelian parent-child relationship for all genotype. Genotype setting. It is important that you consider the genotype setting carefully. Often, you will want to adjust these settings to test the different genetic assumption. The two main panels in the middle of the filter set the criteria for filtering genotype in case on the left and on control on the right. If you do not have control sequence, the right side will be inactive. In a typical scenario, you will be interested in genotype that occur in your case and not in your control. For this type of filtering, the case genotype will be set to keep in the pull-down and the control sample will be set to exclude. You can further control the compound heterozygote definition by opening the corresponding setting control panel. At the bottom of the genetic analysis filter is a setting that lets you filter result based on the sample occurrence frequency of a variant or gene relative to the total number of samples. The sample frequency can be used for different purposes, but generally speaking, for study involving large number of samples, the sample frequency setting can be used to filter out variant or variant within gene that occur at frequency below the level of interest across the sample.